Hi, everyone. We're just going to wait a few minutes to allow folks to join the session. Let's get started. Hello, everyone. Thank you for joining the GenTAC Alliance for our webinar entitled Genetic Variation in BAV and its Role in Aortic Stenosis and Aneurysmal Disease. I am Josephine Grima, and I'm the Chief Science Officer for the Foundation. Oops. So for this webinar, we will use the Zoom's Q&A functionality to field any questions that you may have about today's presentation. So please select the Q&A tab on your screen to submit your questions anytime during the webinar, and we'll cover as many as possible at the end of the session. We ask that you don't use the chat because sometimes those questions get missed there. Also, this webinar is being recorded and will be posted to the YouTube, Marfan Foundation's YouTube channel and the GenTAC Alliance website in the next several days. So today I am honored to introduce our speaker. Dr. Simon Bodie is a clinically retired anesthesiologist and professor of anesthesiology at Boston University School of Medicine. His ongoing research concentrates on bicuspid aortic valve and clinical consequences. He leads the International Bicuspid Aortic Valve Consortium, or BAVCON. BAVCON is comprised of 12 institutions from Europe, Canada, and the United States that have contributed detailed phenotyping and blood and saliva for genotyping. Its members have contributed more than 21 publications to the literature. BAVCON's overarching goal is to identify genetic associations with BAV and its consequence morbidity and mortality. Along with GenTAC and 7 other sites and consortia, BAVCON has identified 36 loci associated with BAV and lately has searched for genetic associations with aortic stenosis and ascending aortic aneurysm in people with BAV. This seminar will discuss the results of these studies. So Dr. Bodie, thank you so much for being with us today. And I'll stop sharing and then you can start sharing and the floor is yours.
Thank you, Joe. I just wanted to say thank you to you and Lauren and the rest of the Marfan's Foundations for this excellent series of seminars and for the invitation, of course. I really enjoy watching them. So let me just share my screen. And go to presentation. Can you see this okay?
Yes, it looks great.
Good, thank you. So as Joe mentioned, this is a group with numerous collaborators, several institutions across at least 2 continents, and numerous other collaborators. I especially want to, at least for this talk, I especially want to thank Sebastian Thoreau and Gina Peloso, who were really at the forefront of the analysis portion of the genetic signals. There are others who've certainly contributed to the patients and the zebrafish work that formed part of the BAV paper, which has been submitted, and we'll see how that goes. So, bicuspid aortic valve disease. I'm sure you know this, but I'll just cover it for the sake of completeness. Incidence of 0.75%. I see numbers from 0.5 to 1.5, but there seems to be a general agreement that about 0.75 is close. Male to female ratio of 2 to 1. Three major anatomical subtypes. Numerous classifications of those subtypes, but just to know that there are three subtypes is important. There's a spectrum of genetic association with BAV from both relatively or very rare variants that seem to be associated with BAV, some family-based work, and this is where the original association with Notch1 came from, and then sporadic disease, which we've concentrated on because it seemed to me years ago that the frequency of BAV was too high to be purely a single gene disorder and too high to be just one or two genes. There's considerable consequent morbidity and mortality. Aortic stenosis. The average BAV person has about an 80% chance of requiring an aortic valve replacement during their lifetime. It's quite high. Sometimes the numbers are as low as 50, but if you follow them for long enough, it seems like just about everyone will end up with an aortic valve replacement of some description. There's a quite high incidence of ascending thoracic aortic disease. Numbers around the 25% mark are common. So it's not a simple disease as it was once portrayed. It's a complex disease with considerable morbidity, and some mortality. There is common genetic variation associated with BAV. That was the first of our null hypotheses when we started the study. It was the first one we embarked upon. We knew there were non-common variants, principally from the Notch1 story, but also folks had been identifying probands and pedigrees with BAV that seemed to follow a good genetic pattern. Some obvious missingness in the genetic pattern, but nevertheless it was there. Recently, Siddharth Prakash has done some excellent work with non-common variants, including copy number variants. So really that's important. We know that there is common, the other null hypothesis was that it was common genetic variation that associates with aortic stenosis and BAV. After all, if 80% of folks get a aortic valve replacement, then there should be. At least that was the null hypothesis. And we figured that the same genetic associations as AS in population controls would also be associated with AS in the BAV cohort. Finally, the last of the hypotheses that we've been examining is this common genetic variation that associates with aortic aneurysm in patients with BAV. The real part of the question was it the same genetic associations as are observed in TAAD in population controls? In other words, those who have a low likelihood of having a bicuspid aortic valve. So those were our three hypotheses for essentially the last 10 years worth of work. You know, it's not a simple thing, the aortic valve. It's got four-dimensional structure. There are variants in the anatomical variation in bicuspid aortic valve. It has numerous both embryologic derivations and also structure in the adult form. So there's lots of individual cells that can contribute to the bicuspid aortic valve and its consequent disease. So we needed to deal with a few issues. First off, discovery of a BAV is usually dependent upon disease presentation of AS, AI, thoracic disease, and usually sometime in the person's 50s. There are certainly folks who have a BAV identified in pre-teenage, teenage, and early adulthood. No question about that. But the majority of folks get it identified somewhere around 50 when they get disease presentation, and then someone does an echo. There's a strong age dependence of disease severity that follows on from the BAV, and a complex inheritance pattern. And as I mentioned, complex embryogenesis. So we thought there was probably strong probability of bias due to other genetic confounders related to clinical presentation. For example, LPA is the strongest signal. Actually, LPA and PALM-D were the strongest signals for tricuspid aortic stenosis. Well known, good story, some good biology overall. And so we thought that the genes that are associated with tricuspid aortic stenosis would be associated with bicuspid aortic stenosis. So we had to account, or felt we had to account for those particular genes. And then fibrillin-1 for TAAD, the very first paper by Scott Lemaire and Diana Melowitz and others. For fibrillin-1, very strong story, and it followed on from the Marfan story very nicely. It came together as a really pretty impressive piece of work. And so we felt very strongly that fibrillin-1 would appear in the list of TAAD genes in BAV. So LPA and aortic stenosis, just to recap on those two genes or two diseases, aortic stenosis. In aortic valve calcium, the strongest, very strong associations, and then PALM-D and LPA. TEX41, not quite as strong, but still a good story. And then for and then further, when was this? And 2016, I think, bicuspid aortic valve was identified as a risk for aortic stenosis. And then the PALM-D, barely significant, or barely non-significant, significant, supposedly, and non-significant. So overall, it was a story that seemed to indicate that bicuspid aortic valve was very, had a common issue for developing aortic stenosis. So this is Scott Lemaire and Diana Melowitz's paper. Here's the fibrillin-1 signal that they observed, very strong, with over the fibrillin-1 gene, some strong characteristics, association characteristics. When you added BAV to the rest, it didn't actually improve anything. If anything, I interpret this as saying that it muddied the signal somewhat. And I'll show you why I think that might be the case in the BAV work. So these were a couple of hypotheses that were generated out of this. We weren't sure about it, so we went on and did the genetics of BAV. When as I said, we've just submitted this paper looking at genetic associations of BAV. Common variants that have already been identified, GATA4 by the group in Michigan, which we contributed, PALM-D, MUC4 from the groups in Leicester and Bonn and Lübeck, have been associated with BAV. We did the same thing, the largest study so far, 9,631 BAV cases among 65,000 participants. So there were about 56,000 population controls in this group, and out of this, through the work of Sebastian and Gina, we identified 36 genomic loci, of which these 4 were included and were the top loci, and 32 of which were novel. We prioritized genes through transcriptomic analysis of aortic valves, done in Laval, and then ran a zebrafish model, 2 types of models, where we confirmed the involvement of the 4 of these novel loci, Wnt4, LEF1, striatin, and CANK2 in BAV valvulogenesis in zebrafish. I admit that it's not necessarily the best biologic model that we can ever come up with, but it was the most straightforward as a screening model. And all 4 of them had good findings. Here's the Bo Yang and Kristen Willer's presentation of GATA4 as being important, and the overall p-value, 10 to the minus 15 for the 10 to the minus 8 and 10 to the minus 8 for another gene near DHX38. We've not been able to replicate this so strongly, but we've certainly been able to replicate the findings for GATA4 or the locus of GATA4. There are other candidates in that region, so even though GATA4 is a very strong candidate, there may be others involved in this. I think GATA4 is a very good story, but it's probably an incomplete story. It accounts for only 2% of heritability, and numerous mouse models occur with GATA4, 5, and 6, knockouts, Notch1, etc. So it's clear that it's not just a single gene, GATA4, that's contributing, or a single pathway. There may well be other genes in pathways. Several other human sequencing and EWAS studies that identified other putative variants, and we thought that mostly GATA4 variants are probably not TAAD or calcific aortic valve stenosis causing variants in patients with BAV. And the other question was, why don't BAV GATA4 variants, or individuals with a variant, have ASDs, which are associated with GATA4, or other outflow tract variants? That's something that we couldn't fit into the model quite so easily. So this is sorry, I'm pausing for a second. Might be out of order. Let me just check. No. So this is the paper done in Lübeck, Bonn, and Leicester. Discovery step, 2,200 BAV patients. Replication step, 421. There was a delay in cardiac development in when you looked at MUC variants in 3Q29. Here's also variants in 8P23, and overall, it seemed to indicate that MUC4, or something in proximity to MUC4, was a, sorry, I've gone backwards, my deepest apologies, had a relationship with BAV. I'm going to guess that there are other genes in this region. There are other candidate genes very proximal to MUC4. This tiny dot down here in there that are related by promoter enhancement. Here are the genes that they identified out of this. Here are the two in MUC4. They're non-coding, and the evidence for this being a long-distance association is, I think, pretty strong. So we did the same sort of thing. More patients, and that's only because of these individuals here, for whom we are very grateful. The Bonn, Marburg, Munich, Leicester cohorts were also included in this analysis, so we felt strongly that we would see an MUC4 signal out of this. There's the Leicester cohort as well, and these other cohorts were also involved. Notably, Gentac was included in this Boston University or Boston cohort because the Gentac samples were analyzed in the same group as the Boston cohorts. So here's what we found. As I mentioned, PALM-D, TEX41, MUC4, and GATA4 were all re-identified, which was reassuring that we'd actually done something right. Here are the four genes that we attempted to replicate by zebrafish experiments, and this appeared to be a good positive finding that these came up. There are others in here for which we really don't have a good handle on. Some of them are candidates, some of them are not. They weren't previously identified. I'll draw your attention to LPA. It's a very, very small signal. It doesn't appear that LPA and the consequent aortic stenosis has a huge effect on presentation. And so it I think it destroyed one of our hypotheses that presentation made a significant difference to how the signals were identified, or how the patients were identified. Similarly, we did not see a TAAD signal out of chromosome 15, fibrillin 1, really almost nothing there. Whereas there are other signals that were actually relatively strong. Let's take LEF1, with a p-value of close to 10 to the minus 20, and others that were also strong, DAOA. So it really looked like there was a good polygenic signal there. And when we estimated heritability, it was about 11.7%, which seems to match pretty closely with BAV heritability. We replicated this also in the Quebec BAV cohort, where a polynomial risk score greater than 90th percentile was associated with an odds ratio of 3.4 approximately with BAV. So a good polygenic risk score signal, but it only came to an AUC of close to 0.7, which implies that it's good, but by no means excellent. Obviously, echocardiograms have an AUC of 1 or close to 1. So this was reassuring, but not necessarily helpful in making any predictive efforts. When we looked at 976 other phenotypes in the UK Biobank, so that was all the phenotypes available, and I want to thank Sebastian for this, Thoreau for this excellent work. It was the polygenic risk score was associated with aortic valve surgery, very strongly aortic valve stenosis, and importantly also a thoracic aortic aneurysm. And I'll show you some of those data. And I've blown it up a little bit here in this part, and I apologize that it's an ugly slide. But here are the top 3, 4, 5 associations of the polygenic risk score with BAV UK Biobank phenotypes, and they all have to do with aortic valve. So very strong and really a very reassuring association. Still complex. When we looked at individually these CANC, ERB4, PRDM6, and striatin associations, you can see the associations here across a smaller region. Very good signals. And when we looked at them in zebrafish, and I won't dwell on this, but here's the normal developmental anatomy in figure form, a normal zebrafish aortic valve or outflow tract valve, and with a left one, Wnt4, 4AB, CANC2, and striatin crispins. We're seeing, these two crispins and these two are MOs. You can see abnormal or relatively abnormal development of the aortic valves. CANC2 wasn't quite so obvious, but it is significantly different. And although this didn't appear in all the morpholinos that we made, or crispins that we made, there was a strong preponderance. Left one wasn't quite so good. It was good, had a highly dysmorphic group. By contrast, Wnt4A and 4B had a lower incidence of dysmorphic aortic valves. And there are just examples of what the crispins and morpholinos look like. So it's all very well knowing that an aortic valve can be bicuspid because of genetic associations. But really this doesn't help us. The first time an echo is done of the aortic valve, it's almost certain that the bicuspid will be identified. So there's really doesn't seem to be a lot of point of having genetic associations except to create a background on which aortic stenosis and thoracic aortic disease can occur. So a good study, but nevertheless difficult to justify in any positives in any value to human beings. So what really counts is aortic stenosis and TAAD. So we took these folks in the study, the genetics component, and looked to see what the morbidity and mortality was. There was about a 5-year lifetime loss from having a BAV. It varied a bit between genders, and it varied obviously across the age spectrum. It didn't kick in until patients were about 55, thereabouts, and So there is some morbidity associated with it and mortality. BAV was independently associated with increased risk and severity of moderate AS and severe AS, relatively good risk ratios, and then obviously more frequent AVR. And this was performed approximately 20 years earlier than the general population. So as we know, it's the single cause of young, and by that I mean under 55, aortic valve replacement. If we see someone with a aortic valve replacement under the age of 55, by far the most common cause is BAV. Over the age of 55, the BAV contribution gets proportionately less as time goes on, or as they age. As part of this, aortic dissection was very rare in BAV. I know we talk about aortic dissection being a lethal disease in BAV, but at least in this group who made it to hospital, I'll bring that up as, dissection may well be a cause of sudden death in BAV, but overall dissection was very rare. It didn't appear to have a significant contribution to that 5 years of lifetime loss. So, genetics of TAA in a BAV cohort. There were 126 previous genetic associations with TAAD. We hypothesized that one or more TAD loci would be observed in a BAV population. So we had two cohorts for this. We had 1,287 TAA cases, thoracic aortic cases, amongst 2,677 BAV participants from Boston and Genentech, because these two groups had excellent phenotyping. And also another group from Laval, Canada, 778 individuals with BAV, and 336 of those had a thoracic aortic aneurysm. In the Boston and Genentech cohorts, TAA was defined as an ascending aortic segment greater than 40 millimeters without Z-score adjustment. Age and sex were adjusted in the genetic model. In the Laval, Canada group, it tended to be a descriptive measure of thoracic aortic aneurysm. And we defined significance of the results as being p-value less than 5 times 10 to the minus 5, not 8, in a locus that had been previously identified as having association with TAAD, or 5 times 10 to the minus 8 in a novel locus. So we're cutting ourselves a break in this, and I'll show you a couple of other places where we cut ourselves a break. And here's the initial or the combined meta-analysis of those two cohorts. And you can see this line should be around here at 5 times 10 to the minus 5. There are 1 or 2 or 3 or 4 loci that at least have one or more SNPs associated with TAD in a BAV population. This I regard as the most significant. These two, I think, are just one-offs, but I might well be wrong. The biology actually supports these. Most importantly, there's no fibrillin-1 signal, much to many folks' disappointment. And so we were concerned by this. The QQ plots appropriately reassuring. And so the summary of that is that no novel loci are observed at the p less than 10 to the minus 8 level. So that was the first of our criteria, or the second of our criteria, and there were no loci that were observed at that. There were 4 pre-existing loci observed at the 5 times 10 to the minus 5 level. And these were CDH13, SLC 25A37, the collagens that are associated with Ehlers-Danlos loss, and HAS2. As I mentioned earlier, there's no signal in fibrillin-1. Then, to try and search not the dregs, but the bottom dwelling fruit, not the top dwelling fruit, we looked at those with p to the less than 10 to the minus 5, and they had a haplotype structure. So they are the folks who it wasn't just a single SNP. It wasn't just a couple of SNPs. They had to have a strong haplotype structure around the signal. But bear in mind, this is not a strong signal. But the haplotype structure was, to enter this list, they had to have more than 10 SNPs around it. They had to occupy a significant portion of the gene or its promoter region. And they had to, what was the last thing? Not be heterogeneous between the two studies. And they had to have some story associated with them. That I could find a set of genes that might have some significance or some biological significance. So here's that group. It's not a group that I would ever like to say these are causative, but I think these represent places to go search, because some of these do have reasonably good stories. And if we want to carry on, an even longer list. These are the ones that are not quite as important or biologically important, although you may recognize a few of them, such as COL8A1 and others. And so I would portray these as having some potential lead into being associated with TAD in a BAV population, but I would not like to say these are associated. Summary: BAV has good inheritance and is a polygenic disease. We are vastly underpowered, it appears, for genetic causes of TAD and AS and BAV. There's literally no signal in a previous gene for AS-BAV or BAV-AS. The genetic signals imply relatively low heritability and a poor predictive value for TAD individuals with BAV. But the prior epidemiology tends to favor there being a modest familial trend for TAD in BAV. And as I mentioned earlier, dissection is rare in BAV. And I'm going to stop sharing, but I'm going to just pull back my, did that work? I'm going to pull back to these genes here and just to the groups that are biased locus identification and share this page, because I have my list here. 1 second while I just attempt to share. And I probably should have put these on the slides. But this, these 4 pre-existing loci at the p times 10 to the minus 5 level, CDH13, a calcium-dependent cell adhesion protein, this was identified both at in the MVP study by Chris O'Donnell, and also James Percelli's study, also here and here. John Eleftherides in his list has this signal for the Ehlers-Danlos vascular type and Ehlers-Danlos types 1 and 2. So I included this in this list. The SLC25A37 mitochondrial iron transporter story, I don't think it's a little more difficult to actually put this out as a story we know much about yet. But nevertheless, I think it could be a good story because it appears in both of these. We don't, as I mentioned, don't see them at genome-wide significance level. So a little tricky, but that's where we stand. I think that's all I want to say, and I will stop my sharing, and then we can carry on. Joe, back in your court.
Thank you. Let me get my video going. Thank you very much for that very informative study. If there are any questions, please put them into the Q&A at the bottom of your screen, and we can certainly discuss those. So I'll wait for a few minutes to see if anybody has any questions. Okay, we have some questions coming in. And the first question is, please discuss the early onset complication patients and families and the recent American Journal of Human Genetics publication. Should we be sending exome genome as the first line of genetic testing rather than panels?
That's a very good question. I'm not sure that that we've reached the stage where we should be doing exome or genome-wide screening as a first-line testing. And I presume you mean for TAAD. Certainly not for BAV. I don't think it adds, I don't think genetic testing will add to the BAV story for an individual patient. Once the BAV is identified, then for that individual, that's the end of the story. And we're not providing any data that says that that you can make a predictive story for TAD or AS in that individual based upon screening, genetic screening or testing. So a mixed story about that. We may well get to the stage where with enough and better genotyping, we may get to the stage where we end up with a TAAD predictive value. I think that's going to be really important because preventing TAAD is important because the morbidity and mortality of it is so high, especially around the time of surgery. For aortic stenosis, I'd like to say aortic stenosis in some way is a less horrendous disease than it was years ago. It certainly appears to be identified more commonly, but it doesn't appear to have the morbidity that, or doesn't have the morbidity that it used to years ago. And that's principally due to TAVR. Also improvements in surgical treatments, but I don't think it's going to make a lot of difference to AS. But back to TAD, there might be an advantage of having some sort of screening, at least to identify targets and at least to identify possible therapies. Right now, there's really nothing that we know prevents TAD in the BAV population. That may change. We may be looking back at this in 20 years and saying that everyone with a BAV with these genes should be on a beta blocker, aspirin, something else.
Okay. And how do you see management of TAA in BAV patients evolving? So you might have just addressed that a little bit, but
yeah, I did. I think I did address that a little bit, but I think it really comes down to we do not have predictive value at the moment. It might occur, but we don't have a therapy for it. If we can end up with some potential drug targets, that would be very useful. I worry about saying that this will be an mRNA treated disease because that will be, at least given the current pharmaceutical climate, that will be very expensive, and only 25% of individuals go on to develop a TAA. And in general, it doesn't appear to make a big difference to mortality. Most of the mortality comes from aortic stenosis associated complications. So I'm scratching my head at this point because I don't think it's going to be, I don't see management changing in the next few years for TAA and BAV. I hope that's answered your question, but it's a very circuitous answer to your question.
all right, thank you. Thank you very much. Are there any other questions? I don't see any others that are coming in at this time. Okay, let me share my screen again. Alrighty, so before you go, we really appreciate if you would take a moment to share your feedback on this webinar by visiting marfan.org /med survey. Your input is really invaluable in helping us to improve our programming. So we would appreciate if you would just take a moment to jot that down and grab that survey. It's really very short. Thank you again for joining us today. And take care. Thank you, Dr. Bodie. Really appreciate your time here.
Thank you.